Start streaming. Okay, so you should be able to see me and hear me. Thank you for bearing with me with that little technical glitch. I'm not exactly sure what had happened on my end, but we are here, we are live. Welcome back to a new year of biotech live workshops. I remain your humble biotech enthusiast, Dr. Danielle Snowflack, and I am the Senior Director of Education at Edvotec. I am so happy to be working with you today to bring biotech back into your classroom, and to kind of give you some tips. Today, I'm super excited because we're going to be talking about an experiment that I actually remember from when I was in high school. We're going to be talking about transformation, and so it's one of those dirty dozen AP bio experiments that we all know and love and have been doing for many, many years. And so I'm going to talk about that experiment, give you some hints, some tips, talk about our new transformation protocol that we're going to start rolling out, and then ways to further extend the learning experience. There, many of you may have been with us before. For those of you who haven't, we are Edvotec. We are the biotechnology education company. So we were founded over 30 years ago now by Dr. Jack Trixian, who was a professor of biotechnology at Georgetown University, biochemistry, sorry, at Georgetown University, which means that the experiment that I actually did in high school might have been an Edvotec transformation kit, which is pretty cool. This era, the last 30 years have been a really exciting time in the biotechnology lab. If we think about it, you get the development of PCR, genetic engineering, transformation. Even the last 5 years have been awesome with the development of CRISPR technologies and other, I personally think the RNA vaccines that we're seeing in the news are pretty cool. So it's just a really exciting time. But at a point, 30 years ago, little of this was being translated into the classrooms, like we're able to do today. And so Edvotec was born to address this need. While our focus is largely on creating kits and equipment for educators, we really want to work with you to demystify science and to foster the next generation of scientists through hands-on active learning activities. But I'll tell you, if you're coming to us from the research side of things, this equipment and the experiments are, durable, they are functional, they are beautifully designed. And so, we're just happy to be giving you the tools you need to do your best in the lab. So before we get to transformation, I just want to bring your attention to a giveaway that we're running. Maria will put the link in the chat box, but if you don't know already, we are giving away an Edge electrophoresis system. That's our new integrated electrophoresis instrument and apparatus that is system that is going to have the electrophoresis chamber, the power supply, and the visualization system all wrapped up into one gorgeous package. And so if you're really looking to supercharge your lab, this is the piece of equipment that you need. It'll also be great for distance learning because you could potentially send it to a student, for home use. And so be sure to enter that contest. And feel free to ask us any questions you may have by emailing us at info@edvotec.com. All right, so without further ado, let's talk about APO8, bacterial transformation. And you guys are probably familiar with this header. This is from the AP manual, right from the College Board. And so this is AP Investigation 8. This is, one of the original AP biology labs. Again, as they're called, the Dirty Dozen. And I personally love this experiment for a couple of reasons. First, it's an awesome and easy way to show that DNA is a primary source of heritable information. So we're passing genes into a bacteria. And to show that DNA is functional there. DNA that we take from one organism can be moved into another. Can be transcribed. It can be into RNA. It can be translated into protein, and that protein is functional in a different organism. In an organism that is not the same as the organism that it came from. So I'm putting the cart ahead of the horse a little bit. But GFP, the green fluorescent protein, is a jellyfish gene, and we can get it to express in bacteria, in plants, in animals. So, this is fascinating to me. And, it's going to be fascinating to your students as well. And this technology is super important for biotechnology as we know it today. In the lab, we can program bacteria to take up genes that make them change color, and that's what we're going to be talking about today. But this technology is used, in industry all the time. So many drugs are produced in living cells in a process called fermentation, which we can use to manufacture products as diverse as biofuels, biopharmaceuticals, and fine chemicals. And so the process of growing and purifying these industrial products and cells is known as bioprocessing. And so basically, we are harnessing cells, we are harnessing life, and turning these cells into factories to make useful products for us in the laboratory. And so once we purify these products from the cells, they can be used for commercial or industrial purposes. So a couple examples that I like to point out are insulin. Insulin, which is a hormone that's used to control diabetes, was the first medication produced for human use using genetic engineering techniques. Biologic medications such as antibiotics, interferons, and blood clotting factors have helped to save millions of lives and improve the quality for millions more. And all of these are produced by introducing genes into cells and using them as living factories. So we're focusing on transformation. Where did it go? Whoops. So we're focusing on transformation, which is a common technique used daily in biotech labs around the world. Let's get back here. By transforming these colors into bacteria, our bacteria we're using is E. coli, we can make it express the proteins that change color. In this case, we're going to be doing the green fluorescent protein, but what you can see is there's a variety in the slide, a variety of different colors that you can do. You can actually have your students do some genetic engineering by inserting genes like the gene that will make the bacteria smell like wintergreen, which I personally like because then they don't stink like E. coli. Anyone who has grown in E. coli culture knows that they are a bit stinky. But it's kind of a fun way to show your students that you introduce a gene, the gene makes a protein, the protein changes something about the cells. And so in this workshop, we're talking about transformation, genetic engineering, and the ways we can expand this experiment to further our student learning. We are recording the demonstration, and the slides are going to be available on our website. If you want to be notified when they are posted, please fill out the form. Maria already put it in the chat box. And so we will send you the slides if you would like. We are also sending out professional development certificates. So if you would like a certificate, please be sure to fill out that form. The form's only going to be available for about an hour after the workshop. So please be sure to complete it by 5 p.m. Eastern time. We want to make sure you're here watching live. All right, so let's talk about plasmids. Let's talk about genes, and let's talk about transformation more in depth. Oh, there we go again. Okay, so in nature, transformation is a natural process that is used to move genes between bacteria. And so in nature, we can do this through two main methods. One is transformation and one is conjugation. And so in transformation, a bacteria is going to take up exogenous DNA, so DNA that's just floating around from the surrounding environment. In contrast, conjugation requires a direct connection between the cells, and it is a transfer of copied DNA from one cell to the other. It goes from your donor cell, and it is transferred into the recipient cell. So in both of these cases, the bacteria have acquired new genetic information that is stable and it is heritable from division to division between bacteria. And so in the lab, we can actually use plasmids to pass genes between bacteria. Let me bring up my camera. So I have a little model here that of a plasmid that we can talk about, and we'll talk about on this slide and the next one. A plasmid in nature, many bacteria are going to possess extra non-essential genes on small circular pieces of DNA, which this is going to model, which is called a plasmid, and that's in addition to their chromosomal DNA. These pieces of DNA allow bacteria to exchange beneficial genes. In 1952, Nobel Prize winner Joshua Lederberg called these plasmids. These plasmids have been used by molecular biologists to study, analyze, and clone genes. They are capable of self-replicating and are distinct from the cell's chromosomal DNA. And although these are not necessarily essential for a cell's survival, an important aspect of the plasmids is the genetic advantage that they can provide to the bacterial cell. So, for example, the gene that codes for beta-lactamase, which is an enzyme that provides antibiotic resistance, can be carried between bacteria on plasmids. And I think I have a beta-lactamase on here. I think so. I also use this sometimes as a little stress doohickey to fiddle with. And I might have taken the ampicillin resistance out of it. But this is our model. You can imagine that there was an ampicillin resistance gene in there. That's the beta-lactamase gene. And that can be carried between bacteria on plasmids. These genes can even be passed between species of bacteria, which leads to bacteria that can be resistant to multiple antibiotics. And so this characteristic can impact the treatment of humans and animals with a given disease. So let's talk about some features of the plasmid. So in the laboratory, we can engineer plasmids to contain genes from different sources. Once transformed into the bacteria, the plasmids turn bacteria into living factories, which create medications, vitamins, and other useful products. For example, scientists use bacteria to make insulin, the medication to treat diabetes. In the classroom, we can program our E. coli with a jellyfish gene, which is going to make them glow bright green. And that one I definitely know is on here because I just saw it. So here's our GFP. And this experiment, again, like I remember transformation from high school about it being a fantastic and really interesting experiment. And I know when your students see them glow green, I'm sure you know that it's super exciting and gets them motivated about learning. So there are 4 main components of a plasmid that are important for it to be used as a way to transfer genes between bacteria. And the ones I'm going to focus on are the origin of replication, or ORI, promoter sequence, a multiple cloning site, and a selectable marker. So the origin of replication is going to be a piece of the plasmid, usually about 50 to 100 base pairs, and that's where replication of the plasmid is going to be initiated. So it's a targeting sequence that lets us know where the that lets the DNA replication machinery know where to start copying a plasmid. It's going to copy the plasmid in both directions, and at the end of the replication, you're going to have 2 plasmids. In reality, in the cell can have hundreds of copies of the plasmid, and each time as the cell divides, it's going to, separate those between the cells. We have a selectable marker. So we can imagine that this is our selectable marker. It looks like it did say AMP at one time. And so that is a gene that's going to allow us to identify transformed bacteria. Antibiotic resistance is often used as a selectable marker, and that gene allows the plasmid to protect bacteria from an antibiotic that would normally kill it, like tetracycline, ampicillin, kanamycin, any of the antibiotics we commonly use in the lab. Our promoter is going to be a DNA sequence. Let's see, here we have a promoter sequence. And that sequence is a DNA sequence that shows RNA polymerase where to start transcribing a gene. And then finally, we do have a multiple cloning site, which is a sequence of DNA that contains several restriction enzyme cut sites. And those are molecular scissors, and they're going to allow us to cut and paste our genes and our DNA of interest into our plasmids quickly and easily, and splice together sequences that are not normally found together in biological systems. And this is super important for biotechnology, medicine, and research. So in nature, your bacteria are going to pick up the DNA from the surrounding environment. In the lab, we can force bacteria like E. coli to take up DNA and be transformed, even though E. coli is not naturally competent. So it's not a bacteria that normally is going to pick up DNA from the surrounding environment. So this can be done with electricity, which is in a process called electrophoresis, or through physical means in a heat shock. And we're going to focus on the heat shock today. In a heat shock transformation, the cells are treated with calcium chloride. So I have some calcium chloride here to show you, which is included with our transformation kits. And it is this salt is going to help make them competent for transformation. We add DNA to the treated cells, and then the suspension is cycled between 2 temperatures. So ice cold, 4 degrees C and 42 degrees C. The charged environment with all of the ions encourages the DNA to stick to the cell wall, And one of the reasons I think transformation is cool is because we still don't know exactly how it's working. But the current hypothesis is that the combination of the charged environment with the salt and the rapid change in temperature, this heat shock, is going to actually change the permeability of the cell wall and the membrane, which kind of allows DNA molecules to be sucked in to the cell where they are then propagated. So, while we have been working really hard on our transformation experiments for years to make sure they're optimized to be robust and reproducible in your classroom, there are still a few tricks to make sure that they work well. I'm going to go through some of the setup and point out places where problems occur and how to combat them. And I'm also going to talk about our new competent cell prep. So we're going to be rolling this out very soon. It kind of gets away from the just pick a colony approach, which you maybe have been using in your lab. This can be tricky if you don't pick the right colony, if your source plates get too old. And this approach is going to allow us to create a solution of fresh cells that can be transformed more efficiently. So the first step I always say when you're getting to do a transformation is you want to make sure that your LB agar plates are prepared and they are, prepared correctly. And so we each Petri plate is going, and I have one that bacteria is on already, but I'll just use this as an example. So each Petri plate contains a solid nutrient agar, kind of feels like a thick jello if you were to put your finger on it and touch it. We use this to culture our microorganisms. Now, pouring plates with Edvotec is really easy. We provide our LB agar as a ready pour, which is the mixture of agar, amino acids, and salts and more, which is the ideal food for growing bacteria. Again, agar is like a science jello. It's solid at room temperature, but it's like it is a liquid when you boil it. And so we're going to microwave the bottle that's included to melt the agarose and then cool it before we pour it. And so we're also making selective media. For example, if we're using our AMP resistance gene on our plasmid, you need to have ampicillin in the plates, right? Because we want that plasmid to be selected for. We want all the cells that don't have the plasmid to be killed. And so we want to put that ampicillin in the media. The problem is that ampicillin is not stable under high temperature. So you've got to make sure to cool that agar to the correct temperature before adding the reagents and pouring your plates. Same with the IPTG. That's going to be our on-off switch for our inducible promoter. Again, you want to make sure that the temperature is right before you add it. The second thing is that you're going to want to follow the specific protocol for your kit so you don't make the wrong types of plates. So we're going to use 3 types of plates in this experiment. We're going to use an LB plate, which has no additives. We are going to use an LB AMP plate, which just has the antibiotic. And then we're going to have an LB AMP IPTG plate, which has our inducer molecule, our antibiotic, and our bacteria food. And so I really like to pour the plates 2 to 7 days before performing the experiments. I pour them at room temperature and I leave them out overnight just on the bench. You're going to ethanol it off first, make sure it's clean, and leave them out overnight. And this allows a lot of the condensation on the lids to evaporate. And this is important because the plates are drier. And when you spread the bacteria on, you're not getting a wet, squishy mess. Like if you've ever done a transformation and put it on a plate that you just poured that day, your colonies can be kind of smeary, they can be diffuse. It's hard to get a good, even smear. You really want to make sure those plates dry. And once you dry them, you can throw them in the fridge and they're good for a couple weeks. You don't have to worry about them once they're in the fridge. But getting them dry is the key to good transformation. All right, so let's talk about our cell prep. So if you've ever made competent cells in the lab, first of all, getting my gloves on now because I probably should have put them on when I was grabbing those bacterial plates. But I'm going to grab them now. And so if you've made competent cells in the lab, our new competent cell preparation is going to be familiar to you. You're going to grow up a culture of cells. So here we have this is our GFP chromogenic host that's included in Bacto bead form with all of our kits. And then here is our Piccolo mini centrifuge. And what we do is we take this culture, we pour it into our 1.5 ml tubes, and then we spin it down. And we want to get a good sized cell pellet. So you can see right here, this white spot here, I had already spun these down beforehand for 5 minutes. And what you can see here is that we have the cell pellet. And I just want to say, if you have any questions while I'm talking through the protocol, please ask them in the chat window, and I will answer them as they come up. Again, this is a new protocol, so it might be something you're unfamiliar with. If you see something you're curious about, let me know. Okay, so we grew this culture, this liquid culture. This is a little different than the plates, the streak plates you might be used to. And so then we've got, oh, my phone is ringing. Decline that. Always mute your phones, especially when you're using them as webcams. So we've got this pellet. What we're going to do is we are going to pour off the supernatant, which is our LB media, and we're going to resuspend it in our calcium chloride. And so in the lab, if you're doing this, you're going to want to keep everything ice cold. That's really important, again, because the temperature is important for the cells to be healthy, for them to, and then for them to want to take up the plasmid. And so what I've done is I've gotten off as much as the LB as I can. And you can see I just have my pellet left in the tube, and I'm going to slowly and gently resuspend by pipetting up and down a few times. Again, in the lab, this would all be done on ice. But what you'll notice is that I'm holding the tube by the top edge. We don't want to grab the tube down here. This is going to warm up your cells, and it's not going to be as conducive for transformation. So you can see I pipetted gently up and down a few times, and I got the cells to be resuspended. You can see it looks cloudy now. I'm going to do that with the next set of cells. Again, you may be used to picking colonies, and that method still works really well. And I'm going to talk about a way that you can do that with your students as kind of an extension activity, to compare and contrast the transformation, the protocols, and see how the transformation efficiency changes. So that's bringing some analytical skills, that's bringing, mathematics into play, forming a hypothesis, which is all super important, especially in the AP experiments, though none of these additions are necessarily just for AP. So I've got my cells in here. I'm going to close it. This is our Piccolo centrifuge. And so if you just have one of these small single speed centrifuges, that is more than enough for, more than enough speed for getting these cells pelleted. I'm going to let these cells pellet now for a few minutes. In general, we would definitely put the cells on ice at this point, but I want you to see what the pellet looks like again when we're done, and just to kind of get an idea of the basic steps. But you resuspend the cells in calcium chloride, you incubate them, which gives them a chance to interact with that salt, which charges the external surfaces, and then we would resuspend the cells in our ice cold solution. You might notice that I am emphasizing cold. And so step 3, again, is to keep everything cold. One of the things that can really go wrong with transformation is that, there are two things, let me step back. There are two things that can really go wrong after the cells are added to the ice cold calcium chloride. One is that the cells are not completely resuspended. And what you could see is that I pipetted up and down a few times to really make sure that the solution was cloudy. There were no large chunks. It was an even color. And that's going to mean that the plasmid won't come into contact with the bacterial cells. The second problem is that the cells get too warm during the resuspension. And this is going to affect that temperature differential that's essential for the heat shock. We recommend resuspending the cells by pipetting up and down gently until no clumps are visible. You don't want to do it vigorously because the cells are going to be fragile when you put them in this hot charged solution, this cold charged solution, and they can be prone to lysis. So if you really pipette up and down, you get really bubbly, that could potentially be a problem for your transformation. In addition, to avoid warming the cells with your hands, you're going to, again, hold the top. Here's another tube for something else, but we're going to just hold the tube at the top. We're not going to hold the tube where the liquid is. And that's going to prevent us from transferring heat from our hands into the liquid. You want to pre-chill all of your reagents before handing them out to your students, and you're going to want them to have crushed ice or an ice water bath to help increase that surface area contact and keep your cells really cold. Our tip 4, add the correct amount of plasmid DNA. So pipetting is a critical skill for your students to have in the accuracy of their measurements, because the accuracy of your measurements can affect everything, especially in biotechnology, where we might be dealing with a single microliter of DNA or of an enzyme. And by pipetting, just 1 microliter extra, you're doubling the amount of reagent that you're putting into your reaction. And so one thing that I found interesting when I was learning more about transformation myself is that the amount of plasmid DNA is critical. So understandably, too little plasmid DNA affects your transformation efficiency. The thing that always surprised me is that adding too much plasmid could also affect your results. And so while no one really knows why, it could be that too much DNA is going to stick to the cells, and that actually serves as a roadblock, and the DNA cannot pass into the cells, through the cell wall and the cell membrane. And so although the plasmid concentration in our experiments has been optimized, pipetting error can affect your students' results. And so you really want to make sure that your students know how to accurately pipette before performing the experiment. And so there are 2 strategies that I recommend for your students. If you are in class, you can practice using our kit S44, which is micropipetting basics. That's going to be a classroom kit. But this is actually a great activity to do if you're in a hybrid schedule where your students are in the lab half the week and they're learning remotely for half the week. We actually have a kit, an Edvotec My Lab kit, where your students can practice pipetting at home. And then it's all done. We actually have a workshop in our live stream catalog where you can watch that experiment being performed. Your students can bring that card in and hand it in as part of their assessment, or they could potentially take a picture, upload it to your e-learning platform. It's really easy to adapt for the remote learning. And so, make sure your students know how to pipette before they get into the biotech lab. And by learning how to pipette, it's going to help them with, their other AP labs, especially the one I'm thinking of, is AP09, which you can do a restriction digest before doing electrophoresis. Okay, tip 5. Oh, sorry, we advanced. Tip 5, time and temperature of the heat shock. Again, this combination of chemical ions and the rapid change in temperature is going to alter the permeability of our bacterial cell wall and membrane. This is going to allow the DNA molecules to enter the cells. And we need to keep that temperature differential in place while we are preparing our reagents for the experiment. So you're going to want to make sure that you have an ice bath, that your cells are on the ice bath, and again, you are handling the reagents in the correct way. You want to keep your cell suspension ice cold. You want to keep your calcium chloride ice cold. And you want to make sure that, the reagents on that end are cold. On the other end, you want to make sure that you have a water bath that can hold the 42°C temperature. I actually like to get it up to temperature, a couple hours before the experiment if you have the time, just to make sure that it stabilizes there. It doesn't get too hot, because if it's too hot, you can actually kill your bacteria during the heat shock. And what we found is that times over 2 minutes of heat shock and temperatures over 45°C actually start to damage the cells so badly that they can't recover. And recovery is the next step of transformation. We're going to let our cells recover we're going to add a nutrient media to the cells. That nutrient media gives the cells, the amino acids, the salts, the food that it needs to start growing and repairing its cell membrane, to start producing the plasmid, and to start doubling, which is what we want. We want our cells to grow and be happy. And so, additionally, during this incubation, the cells are going to start expressing our selective marker. So in our case, it's our antibiotic resistance gene, which is going to let them live on our antibiotic plates. So after 30 minutes of recovery, though, the bacteria will start to double, right? So the doubling time of E. coli is about 30 minutes. And this means that half of our transformants would come from just normal cell doubling. So the amount of time that you recover your cells for is important. We want to give them enough time to start building up what they need to build up, that antibiotic resistance, but we don't want them to sit in solution long enough that they start to double. because these factors are going to affect our transformation efficiency, which is the calculation that we do to see how many cells took up our plasmid DNA. All right, so let's talk about the heat shock itself. So I'm going to just stop my centrifuge now. We'll let it slow down. We never want to stick our fingers in here before it's done spinning, because that could injure your fingers. And so let me get this open. So what you'll see is here are my cells again, and here is the cell pellet. You should see a nice clean white pellet. If you start to see like brown or black, or if it looks sort of sticky, the cells might be lysing or dying. We want to see a nice clean white pellet. At this point, we are then going to add our, add our competency solution. If we're making the competent cells in the lab, again, we're going to add our competency solution. Let me just get this in here so you can see what I'm doing. You would add the competency solution, and you would then, again, repipette up and down in order to resuspend those cells. Again, gently holding the top of the tube as to not heat things up. And then we are going to add our calcium chloride and transform into these cells. Okay. You can see again, there are no large chunks. I try to pipette up and down as little as possible. And again, I am holding the top of the tube to prevent the temperature change. We add the DNA to our cells and then we incubate them on ice for 10 minutes. And again, that's allowing our DNA to stick to the outside of the cells. We're going to place our transformation tubes in the water bath, heat shock it, return the tubes to ice. So that transition between that warm and that cold is what is opening up the pores in the membrane and allowing the DNA to enter. We then add recovery broth to our tubes. We gently flick the tube to mix because we want to make sure our cells are mixing with that nutrient broth. And then we're going to incubate the cells at 37. Again, if you don't have 2 water baths, what I like to do is take a beaker of water, put it in my regular incubator, allow that to get to 37 degrees, so overnight, and then you can put these tubes in a floaty and float them in the incubator. After the transformation is done, we spread the cells on our selective agar media, and then we're going to incubate them at overnight at 37 degrees. If you don't have access to an incubator, you can take the cells. You're always going to want to incubate the plate with the agar media at the top, and the plate, the lid on the bottom. That is going to prevent any condensation from building up on the plate. Too much liquid, again, that can make our colonies smeary. But again, that's a reason why you want really dry plates, because when you turn them over, you can imagine you get all that leaking down into the lid, and that's going to affect your results. So what do the results look like? Oh, well, actually, let's talk about what we're looking for here, what our gene expression is going to look like. Once we get the plasmid into our cells, what's happening? And so we're controlling our genes using an inducible promoter. I like to think of it as a genetic on-off switch. Once we plate our cells on the bacteria, those plates contain, we plate our bacteria on our selective media. Those plates contain ampicillin, which allows us to choose for our transformed bacteria. And some of the plates are going to contain a chemical called IPTG, which turns on the lac operon in bacteria. So a molecular on-off switch, I like to think about it as. If it's there, your gene activation is turned on. If it is not, gene expression is turned off. And so expression of GFP in our plasmid, again, is going to be controlled by our promoter, our inducible promoter. And in this case, we're using the T7 promoter, which is a promoter that came, which is a promoter that has been genetically engineered to be in our bacterial genome. And so this is a special RNA polymerase, which is controlled by the host bacteria's endogenous lac promoter. Under normal circumstances, the bacteria make a protein called lac repressor, which binds to the promoter and blocks expression of this T7 polymerase. Without T7 polymerase, the chromogenic protein cannot be expressed in the cells, the fluorescent protein cannot be expressed, and the cells will not fluoresce, because we need the T7 polymerase to recognize the T7 promoter in our plasmid and turn the gene on, okay? So this T7 promoter is actually taken from a bacteriophage. So it's not native to the bacteria. So this whole signal, I am not sure why. I should be back now. Can you guys see me? Sorry about that. I just went blank for a little bit. If you let me know what is the last thing you just saw. Sorry about that. You know, the internet sometimes goes off, I guess. Let me bring back my camera. All right. So sorry about that. It looks like I lost a few people. I'm super sorry about that. But we're back to talk a little bit. Okay, I'm back with an echo. Let's see. I should only be broadcasting from my microphone and not from my webcam. We're going to see if, okay, back to normal. Thank you. How much did you hear about inducible promoters? Should I go back and review inducible promoters? You hear me fine? Okay, fantastic. Sorry about that. You know, this is live. Tech problems happen. So thank you for bearing with me. So let me just go back. I'll do just, I don't know how much you heard on this last slide. Let me just talk a little bit about the inducible promoter again. Again, it's a genetic on-off switch. One of the things I think is so cool about the inducible promoter is that it's actually a promoter it's a promoter and a polymerase system taken from bacteriophage T7. And we can use this as a genetic on-off switch. So basically, we only express the GFP gene when we want it. And one thing, again, that's super cool about this whole system is that we have this plasmid, it's a bacterial plasmid. It has a promoter from a bacteriophage, a virus, and it has a gene from a jellyfish. So we're able to take this DNA from these different places, these different sources, and get them into one system. All right. So results of our transformation experiment. So here's a schematic of our transformation. We do the transformation with a control. So these cells are heat shocked, but no DNA is added. And we use that to compare to our DNA transformation. So that's going to be the difference between our plus DNA and our minus DNA. So since our bacteria with the DNA are transformed with a plasmid containing a selectable marker and plated both on selective and non-selective agar, we will observe very different results on each of the 4 plates. So our non-selective agar plates will allow for both transformed and untransformed bacteria to grow, which is going to give us a bacterial lawn. When we plate the cells only on media that contains ampicillin, we're not going to see growth from the cells that have not been exposed to the DNA because we don't have the antibiotic resistance to protect those cells. When you plate the cells on the selective agar plate, only the transformed cells can grow. And plus DNA cells should grow no problem on the plus amp plates. But if you only have ampicillin, we're going to see white colonies because our promoter is still not turned on. So we're not activating that green protein gene. On the plus amp plus IPTG plates, our inducible promoter is turned on and our cells glow green. And I have some cells here that I am going to show you because show is always better than tell, right? So I have a couple plates here. And let me get this turned on for you. Oop, wrong way. And so this is using our TrueBlue 2. This is our blue light transilluminator. You can use a handheld UV light if you have it at home to see the cells. If you're using CyberSafe in the lab though, and you have a blue light transilluminator, super easy way to visualize the cells, and it's really easy to get a good cell phone picture of the cells. I know for lab notebooks they're great, for students who just want to show off their pictures, for Instagramming, Twitter, you get a good picture. And so I always think this looks great. So now we have these cells. And one important calculation that we need to do from these cells is going to be our transformation efficiency. I'm going to turn this off for a minute while we talk about transformation efficiency so that I can show you how I'm doing the counting and doing the calculation. So here's my card, here's my calculation. I did the calculations on paper, so I didn't have to try and work them out, while we were here discussing. So transformation efficiency is a critical concept in transformation, and it's a nice way to bring mathematics into your lessons. If you're not already doing it, I recommend having your students try and calculate the transformation efficiency. So this is a concept that is going to allow us to determine how effective our transformation was. So in practice, transformation is highly inefficient. Only about 1 in every 10,000 cells is going to incorporate our plasmid DNA. However, since many cells are used in a transformation experiment, so in our cell pellet, where'd you go? In our cell pellet here, while it looks like a small pellet, maybe the size of a match head, that's going to contain 1 billion cells or more. And so we only need a few cells to be able to take up that plasmid to have a positive outcome. And so because each colony originates from a single transformed cell, we can calculate the transformation efficiency, or the number of cells transformed per microgram of plasmid DNA. And again, this is why it's so important to not allow the cells to incubate too long, because it would affect our transformation efficiency calculation. So if we allow our cells to incubate in the media for 30 minutes instead of 10 minutes, again, that is giving us one cycle of E. coli doubling, because they double approximately every 30 minutes at 37 degrees C. And it would look like more cells have been transformed than actually have been transformed. And again, this is because of cell division, not because of transformation. I just knocked over my LB disposal dish. No big deal. I will take care of that later. So we're going to calculate the transformation efficiency per microgram of plasmid DNA. I have one calculation up here on the screen. If we have 10 nanograms of plasmid to transform 1 milliliter of cells, 1 milliliter cells, and we plated 100 microliters of that mixture and got 100 colonies, then there must have been 1,000 bacteria that were transformed in that 1 ml mixture. Dividing 1,000 transformants by 0.1 microgram of DNA means the transformation efficiency was about 1 times 10 to the 5th transformed cells per microgram of plasmid DNA. Transformation efficiency generally ranges from about 10 to the 4 to 10 to the 8 cells transformed per microgram of plasmid. And so here we're going to see we have my transformation plate. This is the plate that I had on this transilluminator for you. And sometimes when you get a really good transformation, it's really hard to count the cells. And so what I actually like to do is I divide my plate up into quadrants, and I use just a plain old Sharpie to mark off the colonies as I count. And I count in one quadrant, placing a dot for every colony that I find. And in this quadrant, I have 132 colonies. Now you can count the entire plate if you want to. I'm kind of using an estimation method where I counted one quadrant and then I'm going to multiply it by 4, and that's going to give me the total number of colonies on the entire plate. We're kind of doing a little estimation. If you have fewer colonies on your plate, that's fine. Count all the colonies. If you have more, it might actually be difficult to do the calculation of transformation efficiency, because you're going to start miscounting your colonies. And so I take my 132 colonies, I multiply them by 4 to give me about 528 colonies. And so again, one colony here is representative of one transformed cell, because we only incubated it for 10 minutes, again, so we didn't have that doubling. So we have our 500, and this is actually, these calculations are described further in the kit literature, but basically we're going to take our 528 transformants. We know that we're using 50 nanograms of plasmid, so 0.05 micrograms of plasmid DNA. And that is the standard concentration, the standard quantity in one of our transformation reactions. And then we know the volume that we recovered and we plated. And so this is all going to be based on what you're doing in the lab, how much you're adding, how much you're transforming, how much you're recovering. And so when we do this calculation, this gives us about 21,000 transformants per microgram of DNA. So that's about 2.1 times 10 to the 4th. And so that's a great number of transformants. It's a little on the low end, but you can see that is still a lot of colonies. We still had a great result. And this is, it'll basically give your students a starting point for the next part of what I think is important for the AP experiment, which is going to be an independent exploration. Wait, did I go too many slides? My slides are a little misordered, but that's okay. Let's talk about independent explorations because I think that's the natural thing to go to next. So what can we do to change our transformation to increase or decrease the efficiency? So after your students do the experiment once following the protocol that is in the literature, what can we do to change things to make their transformation more efficient or even make it less efficient? So, we are going to want our students to come up with a hypothesis and figure out how to test it. And so, as I mentioned, the amount of DNA is important for transformation. So we can change the amount of DNA and see how that's going to affect our transformation efficiency. You can change the time and temperature of the heat shock. Have your students try and kill the cells and see if you get anything out at the end. These are all great questions for your students to explore. One thing that I think is cool is that you can actually change the salt used for transformation. So it's not included with the Edvotec kit, but there are protocols online for using rubidium chloride instead of calcium chloride for your transformation. And my understanding is that that's actually a slightly more efficient salt to use for transformation. So ask your chemistry teacher. You may be the chemistry teacher as well as the bio teacher. See if there's any of that around that your students can try. They can even try other chlorides, other salts, and to determine the effect, whether that would allow the cells to be transformed. And then you can check the difference between our competent cell protocol and the picking a colony off of a plate protocol. So how do these different protocols affect our transformation efficiency? One thing pointed out in the AP manual that I do like is what effect do mutations have on gene expression? So if we expose our cells or if we expose our plasmid to mutagens before doing the transformation, what happens? So do we affect the plasmid? Does the GFP gene change? Is our transformation efficiency lower or higher? Do we express as much protein? Do we get as bright green cells? These are all great questions for your students to explore. One thing that you can do if you get our 224 kit along with the 223 AP08 kit is that you can try transforming with multiple plasmids. And so the reason I like suggesting those kits is because 223 AP08 includes the GFP protein. 224 uses chromogenic proteins. So we'd be able to see whether we can visibly see one color, like purple, and then turn the fluorescent light on. Do we get a fluorescent colony that is also purple? So can a bacteria hold two different plasmids? And then another question, does the plasmid give a selective advantage over untransformed bacteria? So if we have bacteria transformed with the plasmid, if we mix it with untransformed bacteria and just put that on LB agar, which cells grow better? And you're going to want to do a serial dilution so that you can make sure to see, get a colony number and to just figure out which grows better on that median, whether they affect one another. So there's a lot of places that your students can go from after doing this transformation experiment. One extension that I like is this colony PCR. And so I'm going to use this plate. And so what I want to just do is bring this up. And I don't know whether we're going to be able to see, but I'm going to try anyway. Oop, wrong way. So what you can see is on our plate, we have a combination. So if you look right here, you can see we have some colonies that are glowing and some colonies that are not. And those colonies that are not glowing are what's called a satellite colony. And a satellite colony is a colony that's going to benefit from the presence of a transformed bacteria. So when your transformed bacteria starts to express that beta-lactamase gene, it's going to, excrete it's going to excrete that protective chemical into the media surrounding it, which is going to give our cells kind of a zone where untransformed cells can grow. So, they might not have the plasmid in it, and we can prove that the plasmid's not there because it's not glowing. But, we do have untransformed cells. And so what we can do is we can actually use the polymerase chain reaction to help us figure that out. And so what we would do is we would pick a colony from this plate. We would pick one colony that's glowing and one colony that's not glowing. So this is a little tricky to do with the camera setup, but you can basically take a pipette tip and remove the individual colonies using a pipette tip. And look, that's so cool. You can see my pipette tip is glowing green, so I picked a GFP colony there. And we're going to analyze it by using the polymerase chain reaction. So you want to take a colony. The polymerase chain reaction is an awesome technique that it's like a DNA photocopier. It's going to allow us to amplify DNA even when we're starting from very small quantities, like the amount of DNA in a single colony on our plate. And so this is a high throughput method that was going to allow us to detect our plasmid using the DNA within the transformed cells as a template. In brief, we take that sterile pipette tip and we collect the cells. We resuspend the cells in a PCR mixture. So we're going to use our PCR Edvo beads. We're going to have our pipette tubes. And we are going to have our DNA primers that are going to target just the GFP gene. And they're actually going to target the E. coli genome as well. So we're going to mix all of these together in a PCR tube, and we're going to amplify. So this PCR mixture contains enzymes, primers, and the polymerase we need to amplify our DNA. The mixture is first heated to 94C, which is going to lyse our cells and denature the DNA for the first round of PCR. And then the PCR proceeds as is normal. And we have a couple other live streams that focus on the mechanics of PCR. If you want to learn more about the PCR process specifically. And what you can see is in this diagram that I have on the screen, it kind of is going to show you a close-up of what it should look like when you're doing colony PCR. So after the PCR, we analyze our samples by agarose gel electrophoresis. Any cells that contain the GFP plasmid will show two bands. So the upper band is going to be the band from our plasmid. The lower band is going to be a housekeeping gene from the E. coli genome, because we're going to have both the E. coli genome and our plasmid DNA present in our sample. And that's going to, in any cells that don't have the plasmid, we're only going to see one band that's coming from the E. coli genome. That is a control letting us know that we got cells into the tube, that the cells were lysed, and that the amplification worked. And so a cool extension is to go to this plate, would be to go to the plus amp plus DNA plate, which remember is going to only select for which is going to allow our transformed cells to grow, but it doesn't turn on our on-off switch, which makes them green. So all cells, whether they're transformed or not, are going to look the same color. We're going to go to that plate, and you can pick a bunch of colonies using colony PCR to determine whether it was transformed or whether it was a satellite colony. And again, those satellite colonies should not have antibiotic resistance. They benefit from the resistance from the transformed bacteria. All right, I'm going to go through this. Another cool extension is to do bioart. So add art to your microbiology experiments. So STEAM education is a huge push in, especially, and I especially like using bacterial art to bring some of this art and creative thinking into the classroom. And we did actually do an entire workshop on this for the ASM Auger Art Contest a few months ago, and that live stream is available on our page. But basically, you resuspend and you grow your bacteria in liquid media, and you create a biopaint. So you can either do this from your transformation plates, or you can do it from Bacto beads. Your students can draw out ideas for things they want to design. And then using a loop or a toothpick, they can trace or draw their designs on a plate. So we smear the biopaint on our agar plate, and then we incubate it to allow the color to develop. So you kind of want to take the cells, after you want to let them incubate for about 24 hours or less in the 37-degree incubator. But then you can put them in the refrigerator and the color will continue to develop over a couple days and it gets really bright and beautiful. And so this is a fun extension. If you want your students to try doing something, fun, creative, and a little bit outside the box. And again, if you want to see some really awesome bio art, not just done with transformed bacteria, but with the palette of bacteria, palette of bacteria that naturally have different colors, check out the ASM Auger Art Contest. So that's cool. And the last thing, and this is really important, is bioethics. So transformation is a simple experiment that's going to allow us to, talk about a really deep topic. So bioethics, the thought behind the experiments that we're doing, whether the applications of genetic engineering in the real world, are ethical, are moral, are just. We can think about this in terms of, genetically modifying foods or people, especially in the light of new technology of CRISPR that was developed, and how we can change genomes. What does genomic information mean once it's sequenced? Who owns that information? What do we do with that information? Is there a right to privacy? And so these are all good debate, good arguments your students can do some research on, from reputable sources, reputable books, and to really think about the implications related to the manipulation of DNA and DNA technology. And how do these new technologies revolutionize genetic engineering and make us reevaluate how we're doing it? And so there are, no real right answers to these questions, but it's a place to have students read literature, form an opinion, and this can be used to write a persuasive essay, which would bring in some Common Core literacy skills into your classroom. And I think it's, even in today's, climate, we're talking about COVID-19, we're talking about the vaccine, let's think about the bioethics of the vaccine and vaccine distribution and testing, this is all, important for your students to think about and really grasp their grasp, as they become scientists themselves. So we're almost done. So let me quickly summarize. If you have any additional questions, put them in the chat window now. I will answer them at the end after I do a quick wrap up. So again, in the lab, transformation is a super common technique that we're going to use to program bacteria to take up genes that make them build biofuels, biopharmaceuticals, find chemicals, or even change colors like we did today. Our new competent cell prep, it's more like making competent cells that are used in the research lab, and it will help your results be more consistent because you're starting with healthier cells that have been grown in liquid suspension. You're not trying to pick up a colony off of a plate, so you're getting a consistent number of cells from student to student. They're healthy and they're growing, so that's a really important innovation which will help your results become more consistent. And then bacterial transformation is a great starting activity. So it is something that can be used to bring advanced biotech, bioethics, bioart, and more into the classroom. Okay, so there's this question, which are the two bands from PCR? So let me go back to that. So we're basically in the PCR experiment, we are amplifying the GFP gene from the plasmid, and we are amplifying a housekeeping gene, which is in the E. coli genome itself. So the top band is going to be the GFP gene from the plasmid. And so when we see that band, so in the second and fourth lane, we can see that larger band, that top band, that is from the GFP gene. And so that lets us know our cells have been transformed with the GFP plasmid. The lower band, which we see in all three lanes, which is lane 2, 3, and 4, that is going to be our gene from our E. coli DNA. So if you're picking a colony, that colony of cells is likely going to be E. coli, and then that will amplify, and it should amplify in all lanes. Now, if you don't see that lower band in your transformation, there are two possibilities. One, the cells were not properly lysed, or two, there's some other contamination that got onto your plate, and the DNA primers don't target anything in its genome. All right, so just to conclude, don't forget to enter the contest. As Maria said, we're only accepting one entry per person. So if you've entered already, thank you for your entry. If you have not yet entered, make sure you do that because we're giving away this awesome new integrated system. And then, to request the presentation, I'm going to be posting the presentation and the slides in the next few days to our website, edvotech.com. If you want me to email you when they're available, please be sure to fill out the form that Maria did put into the chat box. And we'll reach out to you in a couple days. Thank you again so much for your time and for the compliment. I'm grateful. These are really fun for me to do. If you have any comments or if you have any experiments you'd like to see, you can put them in that Google form as well. I'm happy to do more AP experiments, happy to do more electrophoresis experiments, but I really want to help you, to see and to learn more, about these biotech experiments and the ways that you can bring them into your classroom. And so thank you so much for your time. We're super active here on social media, both YouTube, Facebook, Twitter, Instagram. So check us out, talk to us. We want to hear how you're doing. If you do the experiment, take a picture and share it with us on social media. If we see it on Facebook or Twitter or Instagram, we do send out special thank yous to our teachers for sharing our results. Because so often, when we get calls, they're generally calling for an order or calling for tech support. But we want to see your good results too. Like we want to celebrate your successes with you. So please let us know. We are super responsive by phone, by email. We just want to make sure that you all have the best possible experience with your science. So thank you. Stay safe. And continue inspiring our students. Thank you for everything you do. Have a fantastic day. Thank you for sticking with me for this hour through the technical difficulties. And we will be back next month with another live stream. So I'm looking forward to seeing you all again. Have a great day, and thank you for joining us.